Alright, so we're going to do a little bit of a video analysis for our Qubit DNA analysis. To start off here, we had pre-prepared samples of both animal blood DNA, which is here on your left, as well as strawberry plant sample DNA. So the dilutions are listed as follows, and they're kind of respective of these tubes here. You have the 20:1 dilution, and then it's factored by 10, so 200, 2,000, 20,000. The same follows here. It was also noted that we had standard 1, ST1, which is on the left, which was around here, and then standard 2, which had a blank in it, and that was like right around here in the sample dilutions because they were used as the controls. For the actual technique, you can see here where the actual samples are being taken from. This is the beginning of the 20 times dilution here from the strawberry DNA, and then the animal DNA is here, and that one was already completed, obviously. So you can see we're going from one to the other here to do the dilution, and we're making sure to mix in between one to the next to properly get the amount of DNA that we need. So this kind of follows through all the way to the fourth one here. So for our Qubit, we're set up here. These are where the PCR, we had special Qubit tubes go. We had used to control our first standard and then put it in and set for the standard 1, which you can see the light here. And then we had taken that out, done it again with standard 2, so that'll be our first 2 readings. Following the setup, you have the option to load multiple tubes, but we'd only done one at a time, and we ran each one individually. This is the actual qubit itself being used. It just reads the sample pretty easily. It tells you the concentration, and you did that across the board for each one of the PCR tubes. And then here's our results. So these are standards. Standard 1 right here. This is actually standard 2. The first standard didn't have a reading because there was no liquid in it. It was just a blank. So standard 2 is here. This is the beginning of our animal DNA. You have the 20 times, 200 times, 2,000 times, and 20,000 times. You can see that the DNA concentration progressively got lower, and then that is across the board as well for the plant, starting at 1.3, 1.13, 0.013, and then it continues to go down. That's actually a pretty solid trend line here for the 10 times dilution. We had also done 2 unknown samples here, and these were the readings for each of those. So in conclusion, we had compared animal blood from cattle to that of a strawberry. Across the board, it seemed that we had a higher concentration of strawberry DNA than in the animals, especially in the 20x solution. So that could be due to error just from the amount of DNA collected in the sample, but it could also be contributed to the fact that strawberries have more chromosomes than cattle do in that sense, so there could be more DNA content in the actual cells themselves. So thank you for watching.